acknowledge that we are and of Matsu's first people and salmon stewards, including the federally recognized tribes of Kanik Tribe, Native Village of Eklutna, Chickaloon Village, and the Native Village of Tyonek. And a huge thank you to the amazing work of these tribes. We heard about a lot of the work that's been going on yesterday. Thank you to everyone else that presented yesterday too. There was so much great information shared, and the jam session, I think I left right before it was really going to turn into a jam session. So I hope that you all had a good time, but it was fun to see everyone smiling and talking and just sharing stories. So I want to say thank you to Jessica Speed for organizing this event. She's over in the corner. If you have questions about the event today or anything going on, feel free to check in with her. So as a long-range planner, I'm just going to reflect a little bit just personally on how yesterday went. So I am a planner. I am not a scientist. So I really appreciated the discussion and conversations we've been having about how to share information, how to find creative ways to bring those science to conservation outcomes forward. So I would love to keep those conversations flowing, and my wheels are turning too. So I see the partnership as a really key tool in facilitating those conversations and connections. And I'm really grateful that I get to be a part of such great work. Yesterday, Freshwater Illustrated also demonstrated how impactful storytelling can be. And I think we can all agree that there are a lot of stories to be told here in the Matsu. So I want to take a couple minutes to reflect on a couple of really specific personal takeaways from yesterday. So the first one, Anna Senecal, you said that it's okay for us to talk about ourselves. So I'm going to take that really literally. I'm going to tell you guys a little story. The second piece of that from Freshwater Illustrated, they had a Jacques Cousteau quote on the presentation: the best way to observe fish is to become a fish. So I'm going to combine those two and tell you just a little story from my childhood about the beginning of my love affair with fish. So hopefully this is a friendly crowd where I can admit that I am not a lifelong Alaskan. I grew up in northern Wisconsin, in the North Woods. It just coming to Alaska has kind of brought me home in a lot of ways. But my dad was a conservation warden there, so I had a really unique opportunity to grow up learning to hunt and fish and explore the outdoors through that lens of conservation and just respect for the resources. So we lived on a lake. I loved the water. I spent evening after evening fishing with my dad, but during the day, I was like a fish in the water. So I had like the really big goggles and snorkel, and I would just float around in the shallows. And my mom and I had to come to an agreement that every 10 minutes I had to move or pop my head up so that she knew that I was still alive. I just wanted to, like, hours and hours, I just wanted to watch the fish and be close to the fish and see the fish. So I was obsessed. I wanted to be a fish. So that was my observation career start there. So imagine my delight when a couple decades later, I was moving to Alaska, coming to this state with like world-class salmon runs, and I really had not had any interaction with salmon in my life up until then. So I still have not experienced salmon with goggles on, so that is a bummer. But I'm hoping maybe someone can help get me out there to do that. But I'm still just in so much awe working to fill my freezer every year. Salmon, it's more than fish. It brings together connection to the land, the waters, the people. You guys are all here because of the salmon, and their journey is epic. So spending these last couple days listening to you guys, connecting with so many of you who share this appreciation, it's fun. So let's continue to learn, share, find creative ways to make a lasting difference for salmon and the ecosystems that they support. So a couple of housekeeping things. The talking room in the back. So if you guys want to chat with people, just try to move out of the room so everyone doesn't have to listen. There is a tidbit sign up at the registration desk. I think there are quite a few folks on there. There may still be a couple of spaces. So one space left. Ooh, who's going to go get it? Hurry. There are evaluations spread out on the tables. Fill them out. You have no excuses. They're right in front of you. Again, I pointed out Jessica if you have questions. We also have Libby over here. If you have not given her your presentation yet for today, bring your thumb drive over to her. There is now hot coffee in the back. If there was anyone wondering why it was cold earlier, that has been fixed. And we might have to wait until later for Aspen Yarrow sang last night at the social gathering, and she wrote a song for this event for all of us. But we have to hold off till a little bit later. Okay. Well, you have to sit in suspense for that. So on this note, I will pass it off to Anna Senecal, who's going to moderate the next session. Thank you, guys.
Hey, thank you so much. Thank you so much. I have to pull this way down. Good morning, everybody. Welcome back. Oh, yesterday we had a great keynote speaker, and today we have an awesome lineup too. We're going to continue this journey in storytelling, and I'm very, very pleased to be able to welcome Dr. Jenifer McIntyre, who came all the way from Washington to be with us in person in our storytelling season. So a little bit about her. She was a member of the team credited with the original discovery of the stormwater pollutant 6-PPD-quinone. And she's here to share with us that notes from that discovery, state of the art, and regulations. So please welcome Dr. McIntyre. Oh, where is it? I don't think it's up back there. So hold on a moment. I can do it. I can do it. Okay. Thanks. There we go. That looks right. Okay, so good morning. My biggest concern today is taking way too long to tell you all the things I really want to share with you today. So hopefully there can be some grace if I start running out of time a little bit towards the end there. My goal is to leave some room for questions, but we might just roll right into our next speaker and then get to ask me questions during the panel. So we'll see how that goes. I am going to try to stick to it a little bit because I am prone to just going off on tangents about the things I'm trying to share and telling stories, side stories related, but I'm going to try to stick to it. So I'm going to go over these 4 things. What is it about the runoff that is acutely lethal? First, the fact that it is acutely lethal for a variety of salmonids, and then leading up to then our discovery of 6-PPD-quinone as a major causal toxicant there. A little bit about what we've learned about why 6-PPD-quinone is toxic, how it's actually toxic, and what we can and are doing about it. So I do want to start, though, by contrasting Matsu Basin with the Puget Sound Basin, where the story of 6-PPD-quinone originates. If we zoom in to the largest population centers in both basins, we can easily see that on the same scale, there's a lot more development in the Seattle area, the Puget Sound Basin, right? No big surprise there. Excuse me. Okay. And when Seattle was first developed, a lot of the streams were channelized, of course, and many were paved over and in fact have never resurfaced. Some, though, have subsequently been daylighted, but more commonly just sections of those streams have been daylighted. And Longfellow Creek here is a classic example of that. Salmon have to swim through a half-mile culvert in the dotted line there before reaching a section that is daylit, and then another upstream section from there is culverted. And yet salmon do come to this stream and attempt to spawn. So with the listing of Chinook salmon under the Endangered Species Act in the 1990s, millions of dollars began being spent on restoring the physical habitat of streams in the urban areas of the Puget Sound area. So this includes riparian areas, removing invasive plants and planting native plants, and then in stream with engineered placement of large woody debris, boulders, and gravel. And I was thinking back about trying to impress upon you like, oh, all of the environmental destruction down in this region, and remembered that my first job in Washington State was actually doing stream restoration and other types of outdoor restoration activities as part of the King County World Conservation Corps, which is now called Earth Corps. That's me on the right there. And Miller Creek, I didn't remember this until I dug up this photo. Miller Creek is a stream I'm going to be talking about a little bit later this morning. And that's where I did some of this work. Now, compared with some urban streams in other parts of the country, like this example from a trip I had recently in Texas, compared with some of these areas, the streams, the urban streams in the Seattle area don't look too bad, right? The daylit portions do look like streams. They do have small riparian areas and natural riverbanks, but they do suffer from a ton of erosion and poor water quality, which brings me to this sad fact here that coho salmon returning in the fall to spawn in urban impacted streams of the Puget Sound Basin are often found to have died before getting the chance to spawn. Fish that are encountered while they're sick, like this one, show symptoms that includes surface swimming, where it's deep enough to, be distinctly swimming at the surface, gaping at the surface, disorientation, and a loss of equilibrium prior to immobility and death. And you can see this is just a gorgeous coho salmon that's hardly even started to turn spawning colors. Now, anecdotally, this has been present since at least the 1980s. And a little side story there is that there's a hatchery in Bellingham that at the time was using a lot of surface water in its hatchery flow-through system. And they knew that if they didn't turn off those surface water inflows to their hatchery when it was raining, their coho salmon would die. And they wrote a white paper, they did a little work trying to understand why, and then that just sort of got buried in the white paper literature. So we finally started studying this problem in the 2000s when post-restoration monitoring brought the problem to our attention. So through years of field studies, we found that the phenomenon is both widespread and recurrent and leads to unsustainably high rates of this pre-spawning mortality. So modeling shows us that even like a gradual increase in this rate of pre-spawning mortality would be expected to lead to local extirpations. This is not a very subtle phenomenon, right? Now, in the real world, salmon populations usually have the benefit of being found as a metapopulation, right? Local populations being linked by, by straying among the basins, right? So in this modeling effort here, we see that if one basin is impacted by this pre-spawning mortality, then you're going to see a serious change in the population of that one basin, but the whole basin as it declines a little bit over time. So the abundances in these remote populations are also declining a little bit. And it's not until all of the basins are impacted that you're going to have complete extirpation in the whole metapopulation. But you can see that effect even across the metapopulation. All right, so pre-spawning mortality is relatively rare in healthy watersheds. When it does occur, it can be caused by a variety of poor water quality conditions, including low dissolved oxygen, elevated temperatures, which can also exacerbate pathogens, of course, or chemical contaminants. And when we first started studying this in the watersheds where coho were dying, sure, we could find evidence of chemical contaminants in the water. We could see metals accumulated in the gills at higher rates than in a pristine watershed. We could see evidence of aromatic hydrocarbons in the bile of the fish, but none of the concentrations that we were seeing looked like they could be responsible for the mortality. Instead, the weight of evidence that we had at the time pointed towards an influx of stormwater runoff coincident with rain events. Some unknown contaminant, or maybe just a combination of contaminants adding together, or maybe even synergism causing this problem. And the reason ultimately that urban stormwater runoff is a problem is because this isn't a forest, right? And so because it's not a forest, it doesn't act like one when it rains. In an undeveloped landscape, most rainwater that doesn't evaporate can infiltrate into the ground, right? Very little of it will flow directly off into surface waters. However, in our built-up landscape, a lot of that landscape is impervious, right? The buildings, sidewalks, parking lots can't usually infiltrate the rainwater. And so as the runoff moves along, it picks up chemicals. It can pull them out of the air. It'll leach them out of building materials. And then by the time that runoff reaches surface waters, it's polluted with a ton of chemicals, right? I have here a time-lapse video showing runoff entering Puget Sound. And this has a little interesting story. Laura James is a diver, and she was out in Puget Sound doing her thing in the near shore, observing all the interesting wildlife. And she saw a column up ahead in the water. She's like, I don't remember there being a structure up there. So she swam closer and closer and realized it wasn't a column, a solid physical column. It was this. And it had been raining, and she was looking at this plume of runoff, and she set up cameras. It goes all night long. So what you're seeing here are those particulates, right, that get carried by the runoff. What you're not seeing are the dissolved chemicals, which are, the source of concern in my research. So one clue that we had as to what could be causing the coho mortalities came from a land use study of Puget Sound watersheds. And the land use most associated with the rate of mortality in a given subbasin didn't correlate best with just imperviousness in general, so development in general, but instead the highest rates of mortality were found associated with watersheds that had the highest amount of traffic in the basin, and the highest amount of roads, and the busier the roads, the higher the rates of mortality. So more than just runoff generally, something specific about roads. So in 2011, I began collecting roadway runoff as I started my postdoc. There was a convenient spot in Seattle along this elevated roadway right here where there was a downspouts into the parking lot at NOAA's Northwest Fisheries Science Center, where I was collaborating at the time. So here's a close-up of the now infamous downspout and then that's a picture I happened to take at the time, a lot of foresight, the very first runoff that I collected. And while runoff, you see it moving along by the, curb of a road or something, looks like mostly just like water. You get a bunch of it together and it looks pretty dark, which now we're like, oh, tire particles. Okay, so we began, though, exposing aquatic animals directly to this runoff, just to try to get a sense of like, okay, well, what could be happening when this water enters when this runoff enters receiving waters or surface waters, right? So in this experimental setup, each of these PVC tubes contains one adult salmon. We aerate the head region of the tube, and then the tube keeps the, what is essentially a large wild fish that's very powerful, calm while we do these experiments. So we found that by experimentally exposing adult coho salmon to roadway runoff, we were able to recreate the acute changes in behavior and the pre-spawning mortality that we were seeing in the urban creeks. So these are obviously just put back into clean water just to observe them after just a few hours of exposure. We found that even small amounts of runoff could kill coho, and sometimes for even very brief exposures, right? The fish would get sick and die after they were moved to clean water even. And that's the white bars here as opposed to the black bars where they died while they were actually in runoff. So overall, this means that stormwater doesn't have to necessarily be a very long pulse into a receiving water if it's at the right concentration in order to trigger a mortality event. We found that the roadway runoff is sufficient to trigger the acute mortality in adults, but also in alevin and juveniles. Basically, all free-swimming life stages were sensitive. And coho are not the only Pacific salmonid that dies from exposure to runoff, but they are the most sensitive so far, we have found. My first species comparison was adult coho and adult chum. And the coho became sick within a few hours of exposure and were all dead by 24 hours of exposure. The chum neither died nor got sick, nor did I see any changes in their blood chemistry that we'd been monitoring at the same time. And juvenile chum were similarly unaffected. In a subsequent experiment just with juveniles, we saw a gradient in sensitivity across these species, with steelhead juveniles being the next most sensitive. Some Chinook died in these exposures, and then sockeye and chum both just didn't seem to care. So that seems important. All right, so what is it in the stormwater that's making the fish sick, right? What contaminants in there that are making them sick? And this was not an easy question to answer. Roadway runoff is very chemically complex. Only kind of recently I guess we're getting on to almost not just recent, but 2017, we published a paper looking at runoff with non-target mass spectrometry and seeing that there were literally thousands of unique chemicals present in these mixtures. We explored the identity of some of those chemicals, but very far from very far from all of them are well understood, and certainly not toxicologically, right? And then finally, the chemicals that we knew about in stormwater, kind of this tip of the iceberg, didn't appear sufficient to cause the mortality that we were seeing. We made mixtures of metals and hydrocarbons, thinking, oh, that's stormwater, right? And couldn't kill the fish, couldn't make them sick, couldn't change their blood chemistry. So clearly, we were missing something in what is the whole mixture of roadway runoff. So what then are the sources of chemicals that get into runoff? Rather than trying to tease through those thousands of chemicals, maybe we could look at it like look at a smaller haystack. So we're getting chemicals coming from particles worn off of tires, from exhaust, from brake wear. We're getting fluids that we intentionally release from our vehicles, like windshield washer fluid, and also things that leak out of our vehicles, right? So which of these are toxic, which are contributing the most to the actual mixture based on their proportions? Reason that it could be easier to find the responsible toxicant if we look in a smaller haystack. So a study that we did used again non-target analysis to look at the chemical mixtures in different sources of vehicle pollution, right? Most of the pollutants on the roadway are going to be coming from vehicles. So in this case, what you're looking at is kind of columns here representing a different unique chemical. We don't necessarily know what it is, but we can tell if it's highly abundant in the dark red all the way down to absent in the dark blue. So we can get kind of a chemical fingerprint here. And then we compared those fingerprints with fingerprints of waters that would kill coho salmon. So different highway runoff samples, different creek waters containing runoff. And then this is the result of a hierarchical cluster analysis that looks at the chemical similarity among those different things. And you didn't see a change in the order here, except that tire wear particles as a source of chemicals clumps down with the waters that kill coho. So this lets us know that a lot of the chemicals in runoff are in fact derived from tires, right? So that's a good clue. However, just because there are a lot of tire chemicals in the runoff didn't have to mean that it was something from tires that was killing the coho, right? To answer that question, we had to ask the fish. So we made a mixture of tire chemicals. I ended up I was targeting a 10 tire mix, but I only got to 9 by the time it was time to do the study. So a bunch of used tires, some new tires, some high end, some low end, just trying to get some sort of representative tires mixture to test here. And then we ran water over those particles so that anything that would just leach into the water would simply do that, would simply move over into the water. And here's this is what a black powder looks like. Some of the earliest tire particles we generated. Anything else I want to say about that? No. Okay. All right. So from our studies with whole stormwater runoff, we know that it has these features, right? It's acutely lethal to coho. It causes certain pathophysiological changes that we've been documenting. And it did not appear to impact chum. So even though they're very closely related species, right? So through a series of experiments, then we compared the response of coho to tire leachate with their response to roadway runoff as a whole mixture, in order to see if we could say that tire leachate was causing a similar kind of toxicity. And in each case, yes, it matched up. The tire leachate was enough to kill coho salmon, cause the same suite of pathophysiological changes, and did not appear to impact chum salmon. And there's the really complicated figure that I published there. I'm like, there's got to be a figure, even though it's such a simple story. So none of the chum died, even in the tire leachate, whereas all the coho died in the tire leachate. All right, so now to isolate the responsible toxicant, we paired the toxicology exposures with high-resolution mass spectrometry. And that was in the lab of Ed Kaloje at the UW Center for Urban Waters. And we made tire leachate again, just by passing water over tire particles. And then we fractionated that leachate and in order to reduce the chemical complexity, right? So for example, some of the first things we did were very simply to put the leachate through a sand filter anything that might be associated with particles. The particles would stick in the sand column. What came out might not be toxic to coho anymore. Well, it was, so it's not associated necessarily with particles. We added what's it called? EDTA, a chelating agent that should remove metals, which would say, if what came out of that no longer killed coho, then it was probably a metal or a metalloid. But that wasn't the case either. So through a very complex series of fractionation steps, we would fractionate, get water samples to test on coho. If it killed coho, we'd try to look at what's in the chemistry. How much simpler did it get? Could we identify what's there? And at the same time, continue to fractionate so we get simpler and simpler mixtures. Now, this approach worked. It didn't have to work. There's a bunch of reasons why it might not have worked, but it did work. So we started with this mixture that contained more than 2,000 chemicals. And each time we fractionated it, we would generate fractions that were not toxic and always one fraction that was toxic. And it would go down, down, down until finally we had a single fraction containing just the presence of 4 chemicals that still kill coho salmon. And it was dominated by this one chemical, C18, H22, N2O2. And we ended up calling that 6-PPD-quinone because although this chemical is not an ingredient in tires, 6-PPD is an ingredient in tires. So it turns out that 6-PPD-quinone is one of the transformation products that's made when ozone attacks 6-PPD. And it does this on purpose. It's put in tires as an anti-ozonant because without it, that's when you get rubber cracking is through various oxidation processes, and ozone's a big important part of that. So you need a chemical there to sort of divert the ozone instead of the ozone attacking the tire polymers and causing that cracking. Instead, it'll attack 6-PPD as a sacrificial molecule, but you end up with 6-PPD-quinone. And this is a chemical that no one had ever reported before. Nobody knew it was there. Okay, I've got a few points here. Let's see, All right, so ozone is continually interacting with the tire surface. So the tire manufacturers put enough 6-PPD in the tires that it will slowly migrate to the surface of the tire over the tire's lifespan and continue to protect the tire, which is why we now have tires that can go, 75,000 miles or something crazy like that, is because of essentially this chemical. All right, another interesting factoid, the bloom you'll get on tires, and sometimes you'll be sold products like, oh, get rid of that like brown color on your tires. That is the transformation products of 6-PPD. And it kind of protects the tire, actually. So you may want to just leave that on there, further slowing the ability of ozone to interact with the tire. But the 6-PPD-quinone can leach from those tire particles as they wear down. And I've got the note here, sidewall versus tread, because you're more likely to see this brown coloration on the sidewall than the tread, because the tread is continuously being worn down, right? And fresh rubber is being revealed. This chemical can also leach just directly out of the tires as they're driven in wet conditions on the road. All right, so we found that 6-PPD-quinone in chemical mixtures like the roadway runoff, like the tire leachate, was able to kill coho salmon at the same concentrations as when we just tested pure 6-PPD-quinone itself, which is the bottom one here. So these are LC50s. So it's the concentration on the x-axis that corresponds with 50% mortality of test organisms. And that's a common way that we'll talk about the relative toxicity of chemicals. You can see that those numbers are pretty much the same, whether we're talking about the chemical in those mixtures or just by itself. So you could pretty much explain all of that mortality that we were seeing by just the presence of this one chemical. So this is why we decided that 6-PPD-quinone is essentially the primary causal toxicant, not a mixture necessarily, even just this one chemical in that mixture. In 2021, a commercial standard became available for 6-PPD-quinone, which allowed us to determine that coho are even more sensitive than we had first calculated. Down with a LC50, that median lethal concentration that can kill half the population of less than 100 nanograms per liter. That's parts per trillion. It's a super, super low number. So how toxic is 6-PPD-quinone? I wanted to know relative to other chemicals that we consider toxic chemicals. So what I did here is I went to chemicals for which we have aquatic life criteria. So these are concentrations in water that are supposed to protect aquatic animals. You can't go higher than this or you might have damage to aquatic animals. And they're based on, some fun math of the most sensitive species that are tested in these tests. So I pulled out from each of those the most sensitive species that had been tested, thinking if coho end up being the most sensitive species for 6-PPD-quinone, how do they rank compared to these other toxic chemicals? And you can see here that 6-PPD-quinone is right near the top of the list, and this is just the top of the top list of aquatic life criteria toxic chemicals. So most of these other chemicals are organochlorides or organophosphates that have a neurotoxic mode of action. And we see that the 6-PPD-quinone is among the most toxic chemicals that we know of for aquatic life. Amazing for a chemical that we just discovered, right? And that has been here for decades. Studies have been published by groups in many places now around the world, showing the presence of 6-PPD-quinone in runoff, in snow, in receiving waters, in dusts along roads, in dusts in your car, in dusts in your house. And the concentrations that are being reported are consistent with what we had shown from the initial samplings that we had done. Often these chemicals are present at concentrations that are above the lethal levels that we've shown for coho. Also, since our publication, additional species have been tested and shown, some of them shown to be sensitive to this chemical. Many, and I've highlighted the ones that were sensitive, many of these fishes I'm showing you here, they couldn't be killed, even at really, really high concentrations. So there's a real diversity and sensitivity to this chemical, even among species that we're considering sensitive because we can kill them with the chemical. And like I said before, closely related species not necessarily being sensitive. All right, it's kind of importantly as an outcome from this is that this problem is not just a coho salmon problem. It's not just limited to the Pacific coast of the United States or the Pacific Northwest even. We have species here found in other parts of North America, and now we're finding species in other parts of the world as well. So that will help us solve the problem is my point. Okay, so how is this toxic? What is happening that this chemical is so toxic? And we still don't actually know, but I'll share a little bit about what we have learned. All right, so one route to learning about how a chemical is toxic might be to understand why some species are more sensitive than other species, right? These differences in sensitivity could be due to differences in the rate at which the chemical is taken up or the rate at which it's metabolized or eliminated, right? And/or it could be like how it interacts at target tissues. So this study here found a new sensitive species, the white-spotted char, and two not sensitive species. And they were the first to show accumulation of the chemical in various tissues of the fish. And they found that the insensitive species accumulated concentrations that were just as high as the fish that had died from exposure to these chemicals. So that couldn't necessarily, the ability to take it up to a certain level in your bodies wasn't able to explain why one species died and another didn't. They were also the first to identify a suspected metabolite of that chemical inside the bodies of the fish, a monohydroxy that they found in various tissues as well. Species differences in toxicity. So is thought to be related maybe to the rate of biotransformation, so the rate at which it's broken down in the bodies of the fish. And so we have that monohydroxy metabolite, which is a phase 1 transformation that our bodies as well do with various chemicals, makes the chemical just a little more water soluble. And then we have this other one, this glucuronidated chemical, which is a phase 2, which makes it much more water soluble. And that's how our bodies more easily excrete chemicals like this. So it could be that the fact that more metabolites are being produced in the insensitive species might mean that the rate at which they can biotransform and get rid of these chemicals explains differences in toxicity. So as the fish are getting sick, we might learn something about insights into toxicity mechanisms. So some of the symptoms that have been observed are these changes in the blood, which I don't personally think is involved, but we can talk more about that later. We also see this progressive impairment in swimming ability, and we see an increased hematocrit. And hematocrit is a measure of blood viscosity, so how much of the blood is cells versus plasma. The blood gets thick in these fish. And that's been shown to be involved in stormwater runoff and in 6-PPD-quinone for a variety of species. So I'm going to talk about these last two symptoms. So using juvenile coho, we were able to track the progress of their illness. And this first departed from controls pretty early. Within the first hour, we see them start to spend more time at the surface of the water. I am taking way too much time, sorry. And then that progresses to the point where they are continuously surface swimming before losing equilibrium and then becoming moribund and dying. So importantly, from this study, we found that fish that had started to swim at the surface, it was already too late. When we moved them to clean water, they died in the same timing and the same rate as fish that just stayed in runoff. So that initial exposure is really important to causing mortality. I'm going to skip this one and talk a little bit about that elevated hematocrit. So in this experiment, we were looking for what's happening as the fish are getting sick. So we exposed the fish to the point where they were losing equilibrium. We injected Evans blue dye into the hearts of the fish and allowed it to circulate through the vascular system of the fish, perfused with saline to get rid of all of the blood. And that's why you can see the gills in this fish are pretty clear tissue colored instead of red. And then we looked for the fluorescence of that dye. Any dye that was left behind would have leaked out of the vasculature into the tissues of the fish. So this is showing you like a head image of the fish, like in this picture here. And you can see a ton of fluorescence, a lot sort of generally, but especially we're concerned about that fluorescence in the brain and what that means. It does mean that the blood-brain barrier is compromised. And that's certainly something to be concerned about. All right, I'm going to skip this one, but I can talk about it later. Finally, we were interested in where's, being able to localize the extent of the leaking in the brain. So this is, what we thought we might see is this sort of punctate staining, it's called, where you see these localized areas where there's leaking into the brain from the vasculature. And the little image on the right shows like around a blood vessel, that there's leaking into the tissues there. So we thought we might see this and be able to say, oh, it's happened, the cerebellum's really important or something. But instead it looked like this. So the leaking is super pervasive and really extreme at the point where the fish are sick enough that they're losing equilibrium like this. Okay, we have some ongoing research looking at the conditions and the life stages at which coho are vulnerable to 6-PPD-quinone. I already mentioned how for stormwater runoff, we found that embryos weren't vulnerable, and we see that again with 6-PPD-quinone, but we do see a delayed impact. Once they do hatch, they start to die. We raised up fish, testing them exposing them as alevin, as fry, and then as par, and we see a progressive increase in sensitivity across these life stages. Perhaps related to yolk absorption being fully absorbed by the time they're par here, perhaps related to other cardiovascular related things. Finally, we did a side-by-side comparison because we knew that adults are vulnerable. We see them dying in the field. We don't see juveniles dying in the field. What is the relative sensitivity of those two life stages? So we did this co-exposure with caged juveniles at the same time and found that, in fact, they have essentially the same sensitivity. So kind of asking this question back again then, okay, well-documented adult mortalities in the field. Are juveniles really dying in stream waters? And this is where I'm bringing you back to the Miller Creek of my youth, of my young adulthood. So this is a pretty small urban creek. This is what it looks like during base flow conditions with pretty low CFS, right? Under storm conditions, we're getting super high peaks in the CFS in this really small basin. And for this experiment, just this past spring, we did an exposure comparing the survival of young of the year coho in 4 replicated flow-through aquaria in 2 different raceways. One raceway received well water into those aquaria. The other raceway received creek water into those aquaria, flow through real time. Both of them had creek water to keep the temperature regime the same in the two exposures. And then we brought in juveniles 3 times across the spring for different storms. So this is cumulative mortality across each of those storms, along with on the right axis, the cumulative precipitation across those storms. And the ADP is the antecedent dry period, so the number of days it was dry ahead of time. So it looks like some combination of those two factors can explain the fact that we're seeing high rates of mortality. This was about 80% mortality for each of these storms. So yeah, juveniles in these systems really are vulnerable, and mortality rates should be considered when modeling population level impacts. All right, I had one little bit here on human impacts, because of course we're all curious, what does that mean for what does that mean for us? Basically, I'm not saying very much about this also because I ran out of time. But yes, we are exposed. Oh, they're saying don't worry about it. Okay, fine. Stop sweating. So yes, we are exposed. Research are detecting both 6-PPD, the parent, and 6-PPD-quinone, the transformation product, in human urine, in cerebral spinal fluid, in plasma. But we're not particularly sensitive, right? We're not dropping like flies when we get to concentrations that we're being exposed to for the most part. And in fact, I kind of made this connection finally. I'm like, oh, look at this number. This is actually 76 nanograms per liter, parts per trillion, which is enough to kill a coho salmon. So we're reaching those concentrations regularly in our bloodstream, even though this is a urine number here. So we're not particularly sensitive, but that doesn't mean that we shouldn't have concern about exposure to this very pervasive chemical. There are a variety of studies with mice now coming out that are suggesting multiple organs may be at risk from chronic exposures, and certainly, especially people living in busier areas, getting exposed to these chemicals through air pollution, for example, this may be a hazard of concern for humans. And so that's not even to mention the other PPDs, the other paraphenylene diamines that are used in products that we are exposed to that are present, now we know, in air, in dust, in soil samples. You can see 6-PPD up here along with a variety of other ones. Okay, the what can we do about it part is, well, one thing we can use is green stormwater infrastructure. And so this is a bunch of technologies that encourage runoff to spread out and to soak into soils where it can be perhaps treated and the chemicals removed, right? So things like permeable pavements, bioretention, we study these at the Washington Stormwater Center. So GSI was originally intended to treat the water quantity problem associated with stormwater runoff, but it usually can also address water quality issues. And in Washington State, bioretention that we have studied a lot is just a simple mixture of sand and compost together. So we have tested the ability of this mixture to prevent mortality of coho exposed to roadway runoff. And these videos are from an experiment where fish, similar to the very first videos like this I showed you, fish were exposed for 24 hours either to clean water or to stormwater runoff in the middle there. We're looking at them 4 hours into the exposure. These fish were going to die just a couple hours later. You can see that almost complete loss of equilibrium. And then the final video here is that same water just passed through that sand compost filter. No more acute mortalities occur there. And we've shown this to be true also for juveniles and for alevin, and for other types of sublethal impacts that we see for other fish that we study exposed to stormwater runoff. However, installing bioretention can be incredibly expensive, particularly in areas that are already very built up, such as the Puget Sound, a lot of the Puget Sound Basin, right? This one I can skip. Get a high lot of, this is a study focused on measuring 6-PPD-quinone in these bioretention systems. It is shown to retain a lot of that chemical. We've looked at other treatment systems as well, other amended bioretentions, compost amended bioswales, which are used alongside highways, for example, that also show good retention of this chemical. And even permeable pavements can have a benefit. This study here, we applied fine tire particles to the surface of permeable concrete in this photo here. Also permeable asphalts exist. And those particles all do make their way into the matrix of the permeable pavement, but not a lot of them make it out. And even better than that is that not a lot of the leached chemicals that would be in the particles that are trapped in those permeable pavements, they don't necessarily come out at a high and same rate as they would. All right. Because of the costliness of GSI, the inability to put it maybe everywhere we need, we also need to consider source control. And this is a conversation we've been having with the tire industries ever since before our discovery. Actually, once we knew it was something about tires, and they were pretty quick to respond after we published our paper about the need to do better, and they've been involved in the process. So the state of Washington does have a roadmap, they're calling it, to dealing with this issue. And as part of that, my lab has started to study other anti-ozonids. Could we find a safer alternative to 6-PPD? Because we're going to need something in tires to protect them from ozone. So initially, we're looking at the relative toxicity of these parent chemicals. And then we're ozonating those chemicals, exposing them to ozone like they would be within the tire matrix, then coming out. And ultimately trying to learn, because something about the structure of these chemicals predict the toxicity. There's little known about that for these chemicals so far. All right, so here's our initial assessment of a variety of these, first tier of potential alternatives to 6-PPD. And note the concentration units here. So this is in micrograms per liter, parts per billion, right? As opposed to the 6-PPD-quinone, the transformation products we're concerned about on a parts per trillion level. So the most toxic of the chemicals that we've studied so far are the double-digit PDs, the 7,7 and 4,4, and whereas the 7-PPD and IPPD appear to be less toxic than 6-PPD-quinone, higher concentrations needed to cause that mortality. So what we did next is we ozonated these chemicals, and we tested them at concentrations much, much lower, so down in sort of the hundreds of, well, actually, this is low micrograms per liter still. But at this level, we did not expect those parent chemicals to cause any mortality. And in fact, they didn't. So that's the bottom picture here. No ozone, no mortality. Those same chemicals then, after we put ozone on them, were now killing the coho salmon. And the toxicity of them as an ozonated mixture of chemicals, of transformation products, and some parent residual still, is in the same order as the toxicity of the parent compounds. So 6 more toxic than 7, more toxic than IPPD. Probably meaning that none of these will move forward as alternatives to 6-PPD. Oops. Okay. Finally, we do now have an aquatic life criterion proposed and being reviewed by EPA in Washington State, that is. And the value that we're proposing is 12 nanograms per liter. Super low. It's going to be super difficult to reach without a lot of changes. But it is a step forward. And the road, of course, is still littered literally with tires and will be for quite some time. So I do want to wrap up now with these thoughts. So tires obviously contain a ton of organic chemicals, and these end up in the air, they end up in the water. We're just starting to learn about where else they end up in terms of, potentially food. And we're going to need both green infrastructure and source controls to address this problem ultimately. And then finally, I hope that this will be, for people who weren't familiar with it, a precautionary tale for the Matsu Basin, hopefully helping emphasize the need that as development of the land continues in this region, that it be healthy and safe for your salmon and your fishes in general. Okay, and with that, I will close and probably not take questions. Okay, well, I'm not going to speak for the organizers, but I'm done. Thank you.
Yeah. Okay, good news, bad news. Bad news, I'm really bad at multitasking and failed to keep time well for you. Good news, we have lots of time for questions. And
Oh, really?
Yes, we do. Yes, I'm so sorry. I'm so sorry. But I'm sure we have lots of questions because that was a fascinating presentation. Thank you for sharing your work. Doc. I know, I felt so bad. Terrible. Fail, epic fail. Questions for the doctor?
You talked about the various methods of dealing with the roadway runoff. getting into the soil and all that kind of stuff to kind of filter it out of the water. Does that stuff eventually break down into non-toxic materials in the soil, or does it just keep accumulating to where at some point the toxicity of the soil itself would become a factor?
Yeah, great question. And for stormwater runoff more broadly, we are concerned about that because things like metals aren't going to break down, right? They're there, they're elements. And so at some point, things like a bioretention, a rain garden, that uses that sort of soil technology, will become polluted enough with metals that something needs to happen to it. It needs to be removed, treated. I would love to see ultimately these systems get mined. Wouldn't that be kind of cool? Remine the metals out of them. I'm not sure that's cost effective, but I'd love for someone to do that analysis. But in terms of the organic chemicals, again, we don't know yet. I'm not aware of somebody who's done this research yet, but it will be relatively simple to do. We know that the PAHs, the aromatic hydrocarbons that get into these systems from stormwater runoff, they do break down. And even though we consider the concentrations in runoff of like a PAH, an aromatic hydrocarbon, to be kind of high, a level of concern for fish, it turns out that in our soil that we use to treat the water, they can start out at higher concentrations. And the fact that we're adding runoff does not increase the concentration of those aromatic hydrocarbons over time because the microbial community likely is breaking them down simultaneously. So I do expect that that will be happening with 6-PPD-quinone as well.
Questions? I'm going to go to the back of the room and then come back to you.
Thank you. Really interesting talk. I was curious, it was interesting to see the blood-brain barrier results with coho. And I was curious if you have any sense or if other folks are looking at like what is physiologically unique about coho that makes them so susceptible. I still am like trying to figure or trying to understand that. Yeah.
Yeah. Well, we don't know yet. That would be nice. The dream lives on to have the answer to that someday. We have done a little bit more work than I showed there, looking at, so the blood-brain barrier is literally just the cells that make the tube, the capillary, that those cells are joined really tightly together, literally with something called tight junctions and other types of proteins that sort of glue those cells together in that tube, whereas other types of capillaries can be a little leaky. They can have a little space between those cells. Blood-brain barrier, they're kind of glued together. So one thing that may be causing a disruption in that barrier would be if those endothelial cells themselves are damaged and, broken or dying. That does not appear to be what's happening. It looks like it's more likely that it's actually the glue between the cells that's breaking down. And it could just be because there are different processes that happen in our bodies that will tell the blood-brain barrier, no, we would like you to open up to allow some process to take place. So it might actually be a direct receptor mediated event. And, in terms of the species sensitivity, we don't know. I mean, there's still one hypothesis. I have a whole adverse outcome pathway that involves like, okay, well, what's before that? It could be triggered by actually an immune response. It could be that coho's immune response is just like wacko over the board too much, right? Kind of like during COVID, I kept thinking like, oh, maybe it's like that, like the cytokine storm, just like triggering too much immune response, then leading to other physiological problems. Anyhow, all speculation.
So coho salmon are pretty much EPA listed as either threatened or endangered all along the west coast of the United States. Do you see if there is some change made with tires that could possibly bring coho back in a big way?
I think so. They're not listed. Ironically, coho salmon are not listed in the Puget Sound region. They're listed as a species of concern, which means they're not actually listed. But then there are populations, other places along the West Coast where they are listed. And particularly in the San Francisco Bay, that giant watershed of crazy human development. I think that this problem is a big reason why somebody in California early on was like, oh, we don't have a problem with our coho salmon. And we're like, that's because you don't have any coho salmon anymore to have a problem with. So I think that it will make a difference for lots of their populations. There's still tons of physical habitat destruction issues and loss of habitat that needs to be countered as well. But in places where we're, rebuilding the habitat, and we're like, okay, if you build it, they will come, and they come, and then they die. You built this attractive nuisance that doesn't help with recovery efforts. So in places like that, it will make a difference.
Testing.
With 6-PPD being like an additive to rubber for anti-cracking and stuff like that, are we seeing it other products other than tires? Like I'm thinking a lot of people have rubber soled waders or like our tough boots that we go into streams with. Like, are we seeing introduction of this chemical from other means?
That's another one I wish I had a really good answer for. A few studies have looked at different types of rubbers. So climbing shoes, for example, yes, those contain 6-PPD-quinone. I don't know where they source the rubber to make the rubber coating on climbing shoes. It could be used tires, right? That's one end of life for tires is recycling of the rubber into other rubber products. So like rubber mats, rubber door mats that are made again from used tires. Yes, those contain it. Trying to remember, Ed's group did a study where they randomly picked some different things, like a bicycle inner tube that didn't have 6-PPD-quinone. Did the tire? I don't know. They maybe didn't test that. Another one was like the sole of a shoe. I think the one that they tested didn't have it in it. But again, depending on the source, it could be like used tire recycling contributing to it. So that's one of the areas that the Washington State Department of Ecology is trying to make inroads. Another road thing. Inroads on is what are all the products that this might be present in? Conveyor belts is one that I heard 6-PPD-quinone, or sorry, 6-PPD is used in. Basically, it's rubber products that need to work well under compression and tension. If it's just a static application of rubber, it might not need an anti-ozonant the way that something that's stretching and compressing while it's in use does.
Great talk. I was, I know we're here about fish, but I saw those concentrations in pregnant women, children. Were those taken downtown Seattle? Was that a general survey? How did those samples get collected? Do I have a ton of 6-PPD-quinone in me?
Okay, that particular study, that was the first study that came out, and it was a study that came from China. A lot of the initial studies looking at human-related impacts have been coming out of China. They apparently have a lot of money to spend on this area of research, which is great, gets us started. I think there might be one. I think there's a cerebral spinal fluid study that was maybe not Chinese. I expect that we're going to find the same things for our populations, at least in denser urban areas. It will be pretty cool ultimately to get a picture of less dense areas, what our exposures look like. How's that for a non-answer?
Other questions?
Yeah, a question from someone on Zoom. Sarah asked, do you know if there have been any documented toxic effects on organisms that have a longer life stage in sediments such as lamprey or mussels?
All right. I know somebody tested lamprey recently, which I was disappointed by because I wanted to test lamprey. Now, I don't know, though, if it was the amicetes or the adults, but they found that they were not sensitive. Other organisms spend more time in the sediment. Aquatic invertebrates that have been tested, again, it's not a long list, but sediment-dwelling aquatic invertebrates that have been tested, not particularly sensitive. There is C. elegans, I'm going to call them a tapeworm. You know what I'm talking about. Those who know what I'm talking about know what I'm talking about. C. elegans. Tubeworm, right? Anyhow, it's a model organism to look at toxicology and things like that. And there is a whole series of papers that's, again, come out of China looking at chronic exposure impacts to that organism, showing a variety of organ systems impacted, kind of like the mice studies. But one endpoint that was impacted at environmentally relevant levels, 100 nanograms per liter, was related to brain function. They saw changes in I'm trying to remember what the actual endpoint is. It had to do with neurodegeneration, though. It looked like it was implicated at those low environmentally relevant concentrations. Will that be relevant across species? Well, very well, maybe.
You touched on doing experiments on different life stages. Did you happened to expose like fertilized eggs to this pollutant? And if so, what were the results of that?
Yeah, I didn't talk very much about that. All right, so I previously, last year I published a paper where I exposed them to just whole stormwater runoff, but it was a situation where eggs were rearing, embryos were rearing in clean water, and I would collect stormwater, bring it out to the hatchery, expose the embryos for 24 hours, switch it back to clean water. Next time it rained, collect more, bring it out. So it was these episodic exposures. And the result of that study was no effect on survival until they hatched. Then they started dying. And prior to that, while they're still embryos, just a little bit of a sublethal impact in terms of they were smaller, various metrics related sort of to growth had been impacted at that point. Now, jump ahead to, we know about 6-PPD-quinone. I have a colleague who did basically the same study, but with pulses of 6-PPD-quinone, saw the same results. And then we coincidentally, when he told me that they had done that study, we were doing that same study that next week. So I was like, how can I make this different? And we started he'd started with eyed eggs. We started right at fertilization, a few little differences, but essentially saw the same thing again. No impact on survival until they hatched. The chorion, and so another thing that we've done this past year that I didn't take time to show is we dechorionated the embryos. So we removed the envelope and saw, is that what's protecting the embryos? The fact that they have the membrane that can, filter out some chemicals. And yes and no, interestingly. So it looks like they were not, they didn't die right away, but they died at the time that they would have hatched is when they started dying, even though they hadn't had the chorion. So the fish that still had their chorion didn't start dying after hatch, right? They didn't have that delayed mortality at all. So somehow they are getting exposed to it through the chorion, but it's not impacting them until around the time of hatch.
Take 2 more questions. I see a hand.
Yeah, thanks for the great talk. I wondered about the density of traffic or the continuity, the concentration, the continual amount of traffic. If you looked at that, because I'm thinking on that map that you showed, we really didn't see I-90, but I'm wondering if there's a little less consistent traffic there, if that kind of affects the amount that gets into the water.
You're talking about the kind of heat map looking one where I talked about land use. I don't remember so you looked and didn't see anything around 90. I mean, a lot of the I-90 stuff is, where's it going? If it goes into Lake Washington, sometimes dilution is the solution to pollution. So I'm not actually sure what I'm answering. I'm sorry. Okay. Well, okay, one thing that has surprised us is we keep calling this like an urban phenomenon. Maybe it's more just a busy road phenomenon. So when I came in here last night, when I flew into Anchorage, I drove out here, I was impressed with the traffic that I encountered. It wasn't nothing, and I definitely made me think, even though the landscape view looks the way it does, as you talk about these localized impacts, that's really what we're talking about is what's coming directly from roads into small enough water bodies that you're going to reach some of these peak concentrations that are a concern. Looks like we have one more question.
Is there somebody that has a burning question? Maybe somebody who's not going to be here for the panel discussion. This is your opportunity. Going once, twice.
Hopefully I'm not preempting Pat's talk with this question, but I was curious if you guys have looked at snowmelt concentrations, because here, especially in Anchorage, they truck snow to these huge snow dumps, and then that lovely snow gets to sit and percolate out into local watersheds. And so I was curious if you've looked at breakdown or attenuation from different times of year.
Okay, and I haven't, and I'm looking forward to
maybe I'm just
what
setting
Pat's
the stage
going to
for
say.
Pat. Okay.
I do want to just anecdotally, I grew up in Ontario, Canada, and snowy winters. And I just hated winter, not just because I was, chilly and inconvenienced by parkas, but because it got to be so ugly if you're in a place with a lot of cars, and you have to get out past that to be like, oh no, it's beautiful. The snow is beautiful. But the way that snow gets so dark and sometimes they say black, probably has a lot to do with settling down of tire particles. So we'll hear more about that very shortly.
Thank you so much. So we have a nice long networking break now. I think there's some snacks in the back. We'll reconvene at 10:45. Help me out. I'm terrible at calendars. Time. Yes, yes, yes. And rest assured, someone else will time keep for the next speaker. Thank you.